Okay, so let's get started. First of all, Happy New Year, everyone. Welcome to our first time in museum for 2026. Um, First, I want to say thank you, everyone, being with us for the whole past 6 years, and then hopefully this year we're gonna have more excellent speakers and also more interesting seminars to share with everyone. So we can get started. Today it's my really pleasure to welcome our first speaker for 2026, Dr. Shao from Shanghai Institute of Material Medica and Chinese Academy of Science. Um, I am going to start with a very brief introduction about Mengle. Mengle currently is a principal investigator at Institute of Medica, Material Medica, Chinese Academy of Science. He received his bachelor degree from Shanghai Jiao University and then continued with his PhD training in Shanghai Institute of Nutrition and Health, Chinese Academy of Science. After he completed a PhD training, he then conducted postdoc research at the University of Texas Southwestern Medical Center in U.S. and then promoted to instructor around 2019. So after a very productive postdoc training period, from 2021, Mengle returned to China and joined Institute of Pasteur of Shanghai, which is now actually converted to or merged into Shanghai Institute of Material Medica, Chinese Academy of Science, as a principal investigator, started his independent research program. So Miller's research is mainly focused in energy metabolism using genetic tools and multi-omics approaches to dissect the physiological functions of complex metabolic regulatory network and their role in different diseases. So his research interests include metabolic plasticity of tissues and organs, small molecule metabolites, and then adipocytes, progenitor cells. Um, his lab actually has several different research directions, cover different variety of different scopes, as I just mentioned, which actually mostly covered the relationship between the metabolic plasticity of peripheral metabolic organs such as adipose tissue and then livers and disease development. Now, the second part of his research direction is physiological regulatory function and mechanisms of action for small molecule metabolites. Last but not least, his group also studying immunometabolic regulatory functions of adult mesenchymal stem cells. Without further ado, I think really appreciate Milo to share your study with us today, and then we are very much looking forward to your talk.
All right. Yeah, okay. Thank you so much for the kind introduction. And once again, I want to thank for this opportunity, especially being the first speaker for this exciting seminar series for the year 2026. And just as Dr. Wu just introduced, my background is majorly surrounding the metabolic regulation. So it's quite an exciting opportunity for me to present to this audience with a very strong immunology background. And hopefully, after today's talk, I will probably can provide some helpful information for just for our thinking regarding the interaction between metabolic and immune systems. So today I'm going to share some recent findings and thinkings about how metabolic signals can regulate immune cell phenotype. So first, a little bit background about metabolism. It's just textbook knowledge that metabolism is one of the essential process for cells, especially for their ability to provide energy substrates and the building blocks of the cells. So just like the every textbook tells us that when the nutrient molecules or other metabolites enter cells, they will be integrated into a very complex cellular metabolic pathway network to provide energy for the cells for their survival. And during that process, it also produced a large number of intracellular metabolites, which are also active bioactive molecules, which have very important cellular functions. And on the other hand, the small molecule metabolites and their regulators can also be very important signaling cascade players by activating the cell surface receptors, and some of the molecules can enter the cells to interact with the intracellular sensors and other proteins to carry out a number of signaling cascade. And both of these two events can eventually translate into the impacts on the very fundamental regulatory mechanisms within the cells. For example, the epigenetic regulation, which relies heavily on the small or metabolite substrate as their as essential components for the biochemical reactions for the epigenetic modulations, and also those uh, metabolic sensors and their activation of their downstream signaling pathways can eventually affect the transcriptional regulation of the cells. So both these two mechanisms are the very fundamental and important mechanisms which can regulate cellular functions and phenotypes. Therefore, the metabolism is deeply involved and contributes to the basic regulations of cell function and their phenotype across a variety of biological settings. So one of a very good examples show that how metabolism or metabolic signals can actually affect cellular phenotype is coming from the example of adipocyte phenotypic switch, especially when people knows that when the animals are exposed to cold, they will induce a very robust adipose tissue remodeling, which is usually called Brownian or Bayesian, in which that the unilocular energy storing white adipocytes will adopt their thermogenic phenotype featured by the expression of marker genes such as UCP1, and their morphology will also be changed into a unilocular and the energy consuming phenotype. So associated with this very strong phenotypic switch of adipocytes, there is also a very robust metabolic reprogramming happening in the cells as well, especially for the purpose to provide sufficient energy for the cells to produce heat. And one of the very critical steps during this metabolic programming is the speed up of TC cycle, which is the major pathway to produce energies. And coupled with this increase in the rate of TC cycle, there's a large number of metabolites concentration will be changed within the cells, which also have regulatory effects, which determines this process of adipocyte phenotypic switch. Oh, I'll just talk about one small example of this. One of the very good examples is about the succinate. Succinate is one of the intermediate of the TCA cycle, just as shown in the textbook. And it was also found to be enriched within the thermogenic fat cells. And the addition of succinate into the cells will boost the thermogenic fat cells' ability to produce heat. And our recent collaboration with Dr. Zhengji Gan and Ting Ting from Nanjing University also provides some additional information about how succinate metabolism could actually contribute to the phenotypic switch of fat cell as well. So Dr. Gan and Dr. Fu just happened to find out that in the adipose tissue, uh, there's increase of the expression of a mitoproteinase LOMP1. Its expression is increased in response to cold exposure in the animal. And when we knock out the LOMP1 gene in the adipocytes specifically, it would block the typical Browning or Beijing phenotype in these animals when they are exposed to cold. That means this protein is required for this process. And then we want to determine whether this process is coming from the effects of the LOMP1 deletion on the phenotypic switch for pre-existing white adipocytes to become beige-like phenotype. So we adapted a genetic linear tracing system, which was developed years ago by Dr. Annabelle Wong and Philip Scheer at UT Southwestern. So basically, in this system, we can inducibly label the pre-existing white adipocytes with a reporter marker gene. Here, we use the GFP as the reporter gene, and then we can trace the fate of these labeled pre-existing fat cells. So basically, to summarize our findings using this system, when the LOMP1 is knocked out in white adipocytes, the cold-induced thermogenic conversion of the white-like cells to beige-like cells is prohibited. Based on the fact that all those unilocular white-like cells in the knockout animals are still labeled with GFP, which meaning they were the pre-existing white adipocytes prior to the cold exposure. That means the LOMP1 knockout actually affects the conversion of those cells to become beige-like. And then we try to find out how LOMP1 does his job in this regulation. And the most straightforward strategy will be just to look for a substrate of LOMP1 as its mitoproteinase property. So through the turbo ID mediated proximity labeling screening, Dr. Gan and Dr. Fu were able to find that the SDHB is one of the protein substrate of LOMP1 in fat cells. And the in vitro protein degradation assay directly proved that LOMP1 can degrade SDHB in an ATP-dependent manner. So just a little bit background about what is SDHB. SDHB is a subunit of the complex 2, which is at the intercourse of the ETC and also the TCA. And this complex is responsible for the conversion of succinate to fumarate. So when you knock out LOMP1 in the adipocytes, in the fat cells, which stabilize SDHB protein levels, it will increase the conversion of succinate into fumarate and decrease the intracellular succinate level in these cells. So then what does it do to actually regulate the thermogenic programming? It is well known that the succinate to alpha Kg ratio is an essential determinant of the activity of a number of alpha Kg dependent dioxygenases, which are responsible for the regulation of the histone methylation levels across a number of genes, including those key genes responsible for the thermogenic programming during the cold exposure, which determines the adipocyte cell fate and phenotype. So in summary, with this LOMP1 dependent succinate metabolism regulation within the fat cells, which can fine-tune the intracellular succinate levels, which eventually talks to the epigenetic regulation within the fat cells, determines their the histone methylation levels across a number of key genes responsible for the phenotypic switch of fat cells. And the metabolic regulation of cell phenotype or cell fate is not only limited to the metabolic organ cell types, it is also relevant to the immune cells. It has been many decades since people recognize that for the immune cells to function, they requires some metabolic uh, substrate to boost their activity, such as for at least 60 years, people realized that uh, immune cells requires glucose metabolism uh, to act their phagocytosis activities. And therefore, the metabolic regulators such as insulin, which will speed up the glucose metabolism in the cells, is essentially critical for some of the activation of immune cells. Here is showing another example that insulin can actually boost the metabolism of glucose and eventually affect the activity or proliferation during the T cell activation. And another very good example showing how a small molecule metabolites can actually regulate the activity of immune cells coming from the findings surrounding another TCE cycle derivatives, itacarnate. So itacarnate is a derivative from the citrate, which is one of the intermediates of the TCE cycle. And when people knock out the enzyme which is responsible for the conversion of citrate to itacarnate, it will release its inhibition ISDH and boost the rate of the glycolysis and TCA cycles, which eventually will activate will increase the immune responses when the macrophages are activated. So therefore, I think there are plenty of evidence showing that metabolism or the metabolic signals are deeply involved in the regulation of cell function and the cell phenotype, including immune cells. So today, I want to try to provide some new evidence that's showing lipid is actually a very important regulator of, at least for a subset of monocyte-derived macrophages during the process of liver regeneration. And this recent work is also a collaborative project between us and two other collaborators in Zhejiang province. And probably they will be the better people to present this story to you. But since I have this opportunity, I would try my best to provide all our thinkings surrounding this topic, especially from a metabolic perspective. So again, a little bit background about liver regeneration. So liver is an organ with extraordinary regenerative capacity. When the liver is injured, either by the surgical damage or drug intoxication, it will start a very strong regenerative response to activate the hepatocyte proliferation to offset the loss of the parenchymal cell in this organ. And during this very complex process, there are two hallmarks which are very relevant. One is of course, it's metabolic reprogramming happened during this process, and another one is the activation of the immune system, both of which are very important for the optimal liver regeneration. So basically, a little bit more details about these two events. So on the metabolic side, there are a few key events such as increased fatty acid oxidation, reduced glycolysis, and increased gluconeogenesis. And on the immune cell side, there's an increase in the immune cell recruitment and activation, and the subsequent production of a number of cytokines and chemokines are also promoting the liver regeneration in the injured livers. So our question is, and the question asked by many is how these two responses can actually interact with each other functionally. So in order to address this question, we focus on two events on either side. The first one is the lipid accumulation in the injured liver. So upon the liver regeneration, there's acute and a very strong mobilization of lipids from adipose tissue. Those lipids will be released from adipose tissue and enter circulation, eventually taken up by the injured liver. There's some evidence showing that this process is actually very important, especially for the acute response to injury for liver regeneration. So we use two models just to confirm and recap this observation. One is the high-fat diet feeding animal model, just to add more lipid during the this liver injury and process. So when we feed animals with high-fat diet for a short period of time, there will be more lipid accumulation in the injured liver. The other one is to use the CD31, which is a primary lipid transporter for liver. So once we block the CD31 with its chemical inhibitor, you will decrease the liver fat accumulation in the injured liver. So when we use these two models and evaluate their effects on the liver regeneration, we'll see there's an increased hepatocyte proliferation associated with the increased lipid accumulation, and there's a decreased hepatocyte proliferation associated with the decrease of the acute lipid accumulation. That means this acute acute lipid mobilization is critical for optimal liver regeneration. So on the other side, we looked into the landscape of immune cells during the liver injury and regeneration. So we use the multicolor flow cytometry and measure the relative abundance of a number of the major immune cell types and look into their temporal changes during 1 to 10 days after the injury. And one of the population attracted our attention is the MDM, whose abundance peaks at the time points which coincides with the time points where hepatocyte proliferation is also at its peak. And then we ask whether the similar question as we asked about the lipid accumulation, whether this increase in the MDM in the injured liver is important. So we use a model to deplete these cells in the injured liver using a CX3CR1 driven genetic elements to drive the expression of DTR to deplete these cells. And after depletion of these cells in the liver, the hepatocyte proliferation rate is much lower compared with the control ones, and also associated with impaired liver regeneration phenotype, which also indicating that these cells are important for the liver regeneration. So now we have two very specific events to study. One is the hepatic lipid accumulation, and the other one is the recruitment of MDM into the injured liver. So the next question is whether these two specific events, which are both important to the optimal liver regeneration, are functionally interacting with each other during this process. So we go back to these two very simple models to evaluate whether they are directly linked to each other functionally. So if you still remember, we use the high-fat diet model and the SSO model again, and to look into the changes in the MDM recruitment. So when there's increased liver accumulation, we saw also an increase of the recruitment of MDM during this process. And on the other hand, when we blocked the influx of lipid into the liver during liver regeneration, the recruitment of MDM seems to be also decreased. So this provided the first piece of evidence showing that there might be a direct link between these two events, which can work together to affect the process of liver regeneration. So based on these observations, we also find something which we found interesting. When we look into the staining of the injured liver, we find a number of the recruited CX3CR1 macrophages are also showing the accumulation, or at least showing the overlapping signals of lipids. Here is a enlarged image. If you can see clearly, here's one of the CX3CR1 positive MDM, and we also find that it is overlapping with the signal of the staining of lipid, which is visualized here by the body P. So this leads to us to ask whether there might be a very direct effects of the lipid accumulation even within those cells, which could be one explanation about the functional interaction between these two events. So next, we use a very routine technology thanks to just thanks to the technically advanced that we can use a single cell RNA-seq technology to look into the cells as the single cell resolution. So we zoom in to check the MDM subpopulation, and they actually can find two major subsets within the MDM in the injured liver. And one of them is even more interesting because its abundance is almost neglectable in the uninjured liver, but its abundance is increased very remarkably after injury, which is consistent with the overall increase of the MDM population during this process. So next, we just use this single cell RNA-seq analysis. We determine two surface markers of these two major subclusters respectively, and based on these two surface markers, we developed a flow cytometry isolation protocol and confirmed the apparent increase of this CD36 MDM, which is corresponding to this cluster 1 here, and it is actually confirmed that their numbers are increasing in the injured liver. And next, we used the flow cytometry to isolate these two newly identified MDM subclusters and compare them with another major macrophage population within the cell, the Kupffer cells, and compare their differences at the transcriptome level. And it is pretty clear that these two newly identified MDM subclusters are quite different from each other, and also are different. Can be distinct a bit. Can be distinguished from the Kupffer cells, which showing their unique features at least at the molecular levels. And then we look into more closely into these cells when the CD36 MDMs compare with their LY6C counterparts. There are a number of pathways related to the lipid metabolism or lipid storage enriched in these cells. And with the microscopic analysis, we can also see the body P staining in these cells, which means there's a significant lipid accumulation in these cells compared with their counterparts. So these two data are direct evidence showing these cells have a lipid-laden phenotype. And on the other hand, we also checked the metabolic pathways enriched in the CD36 MDMs compared with the Kupffer cells, which are usually considered to be responsible for some of the regenerative inflammatory responses during liver regeneration. And even compared with the Kupffer cells, there are some pathways related to pro-inflammatory responses enriched in the CD36 MDMs. And one of the genes I want to highlight here is the expression of IL-6. IL-6 has been known to be one of the key pro-inflammatory factors which promote liver regeneration in the injured livers. And its expression, at least based on our single-cell RNA-seq, is highly enriched within the MDMs, and especially within the cluster 1, the CD36 MDM subpopulations, as the data showing here. And when we compare the expression between the expression of IL-6 between the three macrophage subpopulations, we can find that the MDM, that the CD36 positive MDM, they have a relatively higher expression of IL-6 compared with their MDM counterparts, or even compared to the Kupffer cells in two different models of liver injury. So based on this analysis, we would like to call this newly found CD36 MDM in the injured livers as a lipo-inflammatory phenotype. So we would like to call it the lipo-inflammatory monocyte-derived macrophages, or LIMS. And to our excitement, we also find some supportive evidence showing the presence of these limb-like cells even in the injured human livers. So the first piece of evidence coming from the staining of the injured human liver, we can find some of the staining of lipid co-localized with the CX3-CL1 macrophages. And on the other hand, these CX3-CL1 positive macrophages are in close proximity to the proliferating hepatocytes, which aligns well with the observation in the mouse and in the mouse models of liver injury. And another more quantitative presence coming from the reanalysis of human injured human liver spatial single cell sequence data set. So based on this reanalyze of this data, we found that there are cells expressing limb-specific markers in close distance to the steatotic hepatocyte, and also they are in close proximity to the hepatocytes expressing the proliferation-related markers. So for now, I think we have got some very clear evidence showing there's a very unique and specific hepatic MDM emerge within the injured liver with a very specific cellular heterogeneous features. So here I want to take just a few moments just to talk about our thinkings of how we look into the cellular heterogeneity and how we assess cellular heterogeneity, especially by means beyond transcriptome. So basically, for now, every people just use single cell RNA-seq. This is a very useful and very useful tool to discover and identify new subpopulation cells and also provide functional indications for their function. But from a few years ago, also collaborating with Bo and other people, we tried to look into the approaches which will provide additional information beyond transcriptome per se. So this is one of our paper published a few years ago when we're studying the cellular heterogeneity within the adipose progenitor cells. We combined both the regular transcriptome analysis, and we also added another layer of proteomic information to this study. And we found out that the protein level information is a good complementary strategy to provide different kind of information and even provide some information which is not so apparent when we only look into the transcriptional transcriptomic information of the cells. Just one very clear evidence here is if you look at the transcriptome levels, those subclusters, subpopulations, they cluster closely to each other based on their anatomic locations. But if you look at the protons, these subcluster, these subpopulations, they clustered into the according to their functional distincts, and there are even more information about how we can understand cellular heterogeneity if we look additionally on the protein levels and find some very important functional genes between different subpopulations, as in this paper. Because it is published, I will not go to very details. I just want to make a sense that additional layers of informations will be helpful for our understanding of cellular heterogeneity, and even to find some functional players for a certain phenotype. So following that, we also tried another strategy based on the mass spec analysis of the lipidome to look into the cellular heterogeneity with the help of Dr. Sui Hanfeng in our institute. So this is just one example showing that we isolated the different adipose progenitors subpopulations from human orbital fat depots and subject them to the analysis of the very sophisticated lipidomic analysis. And we can also see that the heterogeneity can be explained at the level of the lipid species. Just showing here that these two populations overall can be distinguished based on their cellular abundance of lipids, and there are some lipid markers specifically enriched in either of the subpopulations of human eye adipose tissue progenitors, which might be functionally related to their phenotype. So based on this, and considering the lipid-laden phenotype of our newly found MDM subpopulations, we play the same trick and apply those cells to the lipidomic analysis here. So in addition to the transcriptional analysis of our hepatic macrophage populations, we also did the lipidomics using the same cell types. And similar to the transcriptome analysis, these three populations of hepatic macrophages can be distinguished based on their cellular lipid contents. And when we look more closely into the lipid species enriched in each of the cell populations, we'll find some marker lipid species enriched in each of these three hepatic macrophage populations. And to the most to the subpopulation we are most interested in, which is the limb or the CD36 MDM, we find the enrichment of a number of ceramide species, which we'll talk more about in the later slides, which can be functionally explain some of its function in the setting of liver generation. So back to the lipid accumulation phenotype of limbs. We use the CD36 as a surface marker for its isolation, for its definition. And if you still remember, we talked in the very first slides that CD36 inhibition is responsible for is actually responsible for the lipid influx of the cells. And we asked the question whether the CD36 or its mediated lipid transport is actually important for the phenotype of limbs. So we knock out the CD36 in these CX3CR1 cells. And we find that either the number of the limbs or their lipid accumulation phenotype is largely abolished when CD36 is knocked out, which means CD36 is essential for this limbs recruitment in injured livers. And once again, we look into the more closely into the lipidomic data of these cells. There's an increase of neutral lipids, sphingomyelin, and ceramides, as I just mentioned, in the CD36 MDM or limbs. And this step is also dependent on the expression of CD36, because when we knock out CD36, the ceramide accumulation within the cells will be largely blocked, including the various the selected species as we showed previously in these cells. And in association of the loss of limb phenotype and numbers, the CD36 knockout animals showed an impaired liver regeneration, as evidenced here by the decreased number of proliferating hepatocytes and the increased liver injury, which also suggests that this CD36 dependent lipid transport step is very important. And how does it affect this liver regeneration? So we look back into the RNA-seq data of our subpopulations, and we found that the UPR response, the unfolded protein response, which usually called UPR, is also enriched in the CD36 MDMs. And despite of the generic ER stress marker, which are not changed dramatically between our subpopulations, there's specific activation of XBP1 splicing, which is indicator of the activation of RU1 XBP1 pathway in these cells. So I think everybody is familiar with RU1 as a unfolded or misfolded protein sensor, and the very key components of the UPR signaling transduction under ER stress condition. But over the years, RU1 has been also identified as a metabolic sensor, which responds to a number of metabolic signals, including lipids. And that activation will actually mediate a large number of downstream biological processes, which are more related to metabolism rather than the classical activation of a stress response. So here if we knock out RU1 in the CX3CR1 expressing macrophages, there's a delayed liver regeneration, which is in line with the activation of the MDM, the RU1 in our limb cells, and these are the functional evidence showing actually the activation is important for the liver regeneration. Then we ask how these two events can integrate together. So here it's a little bit complicated. Just take my words here for the sake of time and simplicity. We set up an in vitro system to mimic the activation of limbs in vivo, basically using a lipid-treated hepatocyte culture and take their conditioned medium to stimulate the monocytes, which could induce the expression of limb-like cell markers, and also the activation of the ceramide metabolism in vivo. So in this system, we use the chemical inhibitors of the ceramide synthesis pathway to block ceramide synthesis and to evaluate whether they will affect the activation of RU1 in these cells. So although these inhibitors does not directly affect the monocyte's ability to acquire the limb-like phenotype, such as the expression of CD36, which is a defining marker of the limbs, the activation of RU1 is actually downregulated. That means the ceramide, the CD36-dependent ceramide increase within the monocytes is suggests to be an important step to activate IU1, which is functionally important uh, for the limbs for the MDM's role in liver regeneration, as shown in the previous slides. That if you knock out IU1, the phenotype is impaired liver regeneration. So then probably the last question of this pathway is what's the down mediator of IU1 pathway, which eventually carry out the functional role of these cells to regeneration. So we compared the four sets of RNA-seq, and we eventually find one last candidate. Sometimes one is the best, right? So and it happens to be IL-6. If you still remember in the previous slides, I talked about IL-6 expression is highly enriched in the MDM population, and especially within this limb population, at least based on our single-cell RNA-seq data. So we now count IU1, we'll see a decrease of IL-6 expression in the whole liver tissue, and also the protein levels of IL-6 is also increased almost by half in the IU1 knockout animals, which means IL-6 is a downstream target of IU1 pathway in these cells. And lastly, we use a IL-6 overexpression plasmid to rescue the expression of IL-6 in the IL-6 in the IU1 knockout animals, and evaluate their phenotype regarding the liver regeneration, and consistent with the model we want to propose. The IL-6 reinstauration can actually rescue or largely correct the defects in the hepatocyte proliferation almost to a level comparable to the control animals. So here comes the summary of this study, that we believe that our evidence has shown a emergence of a MDM subset whose recruitment is dependent on the acute lipid influx into liver during liver injury, and promote the regenerative response of liver. And this I think we showed some a new evidence and new information about how lipid metabolism can interplay with the immune remodeling in a very physiologically related setting, even in both rodents and human. With that, I want to close with the acknowledgements, especially for our collaborators, for the liver stories, Bo and Ying, and for the previous data I showed. The long point story is a collaboration between Zhenji and Ting, and I really appreciate the efforts Sui Han has inputted into set up the pipeline for the lipidomic analysis of a small number of cells. And also thanks to the other collaborators, both in China and the United States. And I thank you for your attention, and I'm glad to answer any questions.